Here's our mRNA strand, which was made in the nucleus off of one strand of DNA. When they split apart, created an mRNA strand with the U instead of T. The purpose of that is so the mRNA can leave the nucleus, go into the cytoplasm, and go to a ribosome. When it moves to the ribosome, it's going to these three bases, and then the next three, and so on and so forth, are called codons. They code for an amino acid. When we meet in the ribosome with tRNA, which is transfer RNA, and rRNA, which is ribosomal RNA, all three of these are going to help determine what the amino acid is, and a long chain of amino acids create a protein, which is a polypeptide chain. Now, we will look at, once we get the chain together, once you have a long chain of amino acids, it's going to, the order in which they're in will code for a specific protein. It depends on the sequence that they're in. But these code for specific proteins, but they can be arranged in different ways. If you want to watch a video on that, you can. Here are the 20. There are 20 amino acids. You'll see those again here in a second. The mRNA strand that we had, that we made in the nucleus off of DNA, that moved in the cytoplasm, and then now is meeting up at the ribosome with rRNA and tRNA, it's going to go through translation. What's happening is it's being decoded into a protein. Again, it's done in the cytoplasm, and it's going to meet up, like I said, with tRNA and rRNA in the ribosome. Here are the three right here. Here's messenger RNA. It's going to be the strand that we made in the nucleus. Here's tRNA. See how it looks like a T? It's transferring. We'll see it transfer over here in this picture here, where it's transferring the anticodon to allow us to code for the amino acid. The rRNA actually comes in and assists in this process by helping put them together. It's a two-piece process there. So it's kind of like a ticker tape. It goes through, feeds through the rRNA in the middle. So you'll have the tRNA come in here. The tRNA will come in. The mRNA will feed through it, and it will help code. The anticodon will help tell what amino acid needs to be picked up. As it continues to take on amino acids, it's going to start making a long chain, which is called a polypeptide. If you are starting an amino acid, will be AUG, is a start codon. So, like I said, the 3. And then you have a stop codon, which is AUC. This is stop, and this is start. So, when you have a start, it means it's going to read the mRNA and keep reading, creating these amino acids, bringing the amino acids in. Every 3 bases are going to be read and coded, and the anticodon is going to tell what amino acid to pick up. Once it gets to this point of making the amino acids, it's going to stop, because that's what the mRNA told it to do. Now, once we have our amino acid chain, our polypeptide chain, it's going to code for something. So if we look here, we're going to read this chart here over here. This is the chart we're going to read. You're going to start from the middle and read out. You're going one at a time. each of your, like here it says, to decode the code, the first letter in the set bases of the center of the circle, which is here, then we're going to move outward all the way out to the outside. So there are 3 stop codons called nonsense codons. What they do is they tell it to stop, no more amino acids. These are your 3. This one here is your start. It tells you to start. So we know our first one's going to be A, U, G, and that is our first amino acid. Then we will have several other amino acids to determine. We'll practice this later, but let's say we get through a full chain of amino acids and then now we're at the end and we're going to stop. So one of these 3 are going to be your stop codon. So it's U, A, G. And there it says stop. So this will tell the mRNA to keep feeding through the ribosomal RNA with the transfer RNA bringing in the anticodons until it comes to this stop codon. Okay, there is one other table that's very similar. You're going to look and see it says first letter, second letter, and third letter. That's exactly how you read it. So again, this one has your start and stop already on it, but let's look. You go to A, U, and G. So see how I'm in the same box? This one is telling me here. And then now I'm going to find the G. Tells me here. So this is where I end up, right in this box, which AUG is our start codon. Now we said that, let's look at this one. It says, we'll say UAG. UAG is our stop codon. Remember, you have a bunch of amino acids in the middle, which we'll practice. But this is the stop codon once we have a whole chain of amino acids. But we're going to look and see. It's U, A, and then G. Which brings us right here, because that's where all of them meet. Think about drawing a line to see how they all intersect. Where do they intersect? Right here at this stop codon. And that is how you read these charts. So we will practice in class. I'll also make a separate video to practice them too. But this is the lecture to end the notes for that. We still have another section to cover, but there is another video down here for you, and hopefully this is helpful.